Everybody from Wednesday lab have your procedure for Dr. Morgan?
Tuesday.
Tuesday lab. That's right.
Tuesday and Wednesday.
Tuesday and Wednesday. I'll get it Okay, we're going to finish up cell respiration today, and that will be the ending point for material for the exam on Friday. So remember that you're supposed to come equipped with any questions you might have on Thursday for review. Otherwise, I'll have some questions for you. Okay, so what I want to do first thing is do a little bit of kind of simulation or modeling of the process of oxidative phosphorylation. What does oxidative phosphorylation refer to? What are we talking about when we talk about oxidative phosphorylation? We're talking about making ATP, okay? And this is an oxygen-dependent process, okay? And which of the three phases of cell respiration are associated with oxidative phosphorylation? Electron transport system, okay? And where's the electron transport system located? The inner membrane of the mitochondria, okay? And what is the purpose of that electron transport chain? Why does it exist? To remove hydrogens from the reduced coenzymes NADH and FADH, okay? And as it removes those electrons, they eventually have to end up somewhere. Where do they end up? They're grabbed by oxygen to make water, okay? And what happens is those electrons are being passed through the electron transport chain on their way to oxygen. Creates a hydrogen ion gradient, and we pump hydrogen ions from here. Where is here? Where are you guys sitting? Matrix of the mitochondria. Out here to this inner membrane space, okay? And then once we build up a high concentration here, what are those hydrogen ions going to want to do? Go back into low concentration, but they kind of go, can't get across that phospholipid bilayer. It's a hydrophobic barrier, they're charged, not going to work. But if there are channel proteins in the membrane, then the hydrogen ions can flow through those channel proteins. And that channel protein is also an enzyme. So what enzyme is it? ATPase, which catalyzes the synthesis of ATP out of ADP and phosphate. Okay? So we've got several different pieces of colored paper out around various places in the room to represent different components or players in this particular process. So what I'm going to do is, I told you I typically like to use this row of chairs for the electron transport chain, the one on the outside here, because of the fact that they are tiered and they go down. That's indicative of what happens in the electron transport chain. We proceed with a decrease in free energy. But I think it's easier to see this row, so I'm going to use this row. Okay, so there are some brown pieces of paper in these particular set of chairs right here in this inner membrane. And remember, this is a phospholipid bilayer. So there's some phospholipids with their polar heads out here, and their hydrophobic tails in here. There are some phospholipids over here with their polar heads and their hydrophobic tails, okay? And we've got two kinds of compounds that make up this electron transport chain, or the electron transport system. The brown pieces of paper are representing molecules that can donate and accept hydrogens. So we've got some here, we've got some here, we've got some here. And those particular compounds are oriented so that they tend to face the inner side of the membrane. So if I really wanted to make this realistically, I'd have those brown paper people push their chairs right back to the back of the stopper, right there. And then the green compounds are compounds that accept and donate only electrons. and they tend to face this side of the membrane. So it's almost like they're sitting there going, gimme, gimme, gimme from here, and these folks are saying, gimme, gimme, gimme from here. Okay? Okay, we've got some oxygen molecules over here. We have some protons over here. Orange represents hydrogens, but this is only part of a hydrogen. This is only the proton, or the positively charged portion of the hydrogen. And then we've got some hydrogens, protons and electrons. We've got some oxidized FAD molecules, and we've got some oxidized NAD molecules. Okay? So those are the major players we're going to deal with right now. So, what set of reactions is happening within the matrix of the mitochondria? Citric acid cycle, right? And what's happening during the citric acid cycle? Well, if you include the transition reaction, which I kind of lump those together, pyruvate has moved from the cytoplasm into the matrix of the mitochondria. What produced the pyruvate? Glycolysis, okay? It came from glucose being converted into 2-pyruvate, okay? And then that pyruvate becomes decarboxylated, it becomes oxidized, it has a coenzyme A added to it, and it forms what compound? Acetyl-CoA. And that's what feeds into the reactions of the citric acid cycle. So what's happening during the citric acid cycle? Well, basically this is what's happening. All the hydrogens are being taken off of pyruvate, and what's left when you take the hydrogens off pyruvate? Pyruvate is basically carbon, hydrogen, and oxygen. Take the hydrogens away, we've got CO2. Okay? So the citric acid cycle is going to be producing CO2, and it's also going to be producing some hydrogens. Okay? Where do those hydrogens go? They go to some of the oxidized coenzymes. And so there's 2 for one of those oxidized coenzymes. There's another 2 from another oxidized coenzyme. There's another 2. And I'll give some to FAD too, because hydrogens are picked up by FAD. Okay? This Krebs cycle keeps hopefully turning round and round and round and spitting out hydrogens, basically. Well, here's 2 more hydrogens. See that? Can't take them, can you? No? Don't have any more oxidized. I should have given you some of the purples right there. So eventually, these hydrogens that are being torn off of those pyruvate molecules don't have anywhere to go. So the reaction stops. Krebs cycle stops. Until we can get some more oxidized NAD available to pick up some more hydrogens that are being liberated. Okay? So that's not good. So we would like to recycle these. So what do? We donate this to the electron transport chain. And this compound can donate a couple of hydrogens to the electron transport chain. Oh, Nathan, we're good to go again. So now you can accept some more from the Krebs cycle, right? But then you got to get rid of them again. Okay, well, did you give them to Ethan? No. You can't take them. Can't do it. Sorry. So what happens? He has to get rid of his. Then he can take more from you, right? So, he tries to give his to Brandon. Brandon's kind of a picky guy. He doesn't want just hydrogens. He can only take the electron portions of these. He's got the electrons now. Now I got these protons sitting here. Are they going to be happy inside of this phospholipid bilayer? No. They're going to tend to be spit out of that phospholipid bilayer. And since he's sitting right here at this face, where's the likely place these are going to be spit out? Out here, into the intermembrane space. Okay? We're accumulating some H pluses here. Okay? Well, give me some more of your reduced NAD. Okay, so you'll take those. Back here again. Okay. Can he get his up? Nope. He's already reduced. So he's going to get rid of his electrons over here. So now he can take more from here, right? And you can give more to here. Okay? But this is a hydrogen carrier. And he said, I got electrons. I need some protons. And I'm sitting here. Give me some protons. Ah, Cody's got some protons. We'll take a couple protons. Where did these come from? Where did these protons come from that are in the matrix? Citric acid cycle didn't produce them. They produce hydrogen. Where did we get protons in the matrix? What's the matrix full of? It's full of water. And what does water tend to spontaneously do? Dissociate. Okay? So there's a supply of protons being produced by the dissociation of water, and those protons are being pulled away from the matrix and donated over here. Okay? So now Ben's happy. He's got his two hydrogens. Okay? So he tries to give them away so he can take more. Kyle's one of those picky guys. And he'll take the electrons and not the protons. So we put some more protons out here. Whoops. Got an extra one there. Okay. And then the same thing is going to happen over here. What do you got in there? Proton for me. And as we donate things down this direction, then we're going to take the electrons here, and we're not going to take the protons. And so as we pass things through the electron transport chain, protons are effectively being taken from the matrix and shoved out on this side of the membrane because of the fact we've got these two different kind of molecules that make up the electron transport chain. The electron donors and acceptors and the hydrogen donors and acceptors. And the fact that they are asymmetrically or unequally distributed, and some face this side and some face this side, kind of ensures that the protons end up accumulating out here. Okay? So we're building up all of these protons here. So what happens? If a pair of proteins goes through this ATPase channel, that provides sufficient energy to make one ATP. Okay? So how many protons did one reduced NAD pump out here? 1, 2, 3, 4, 5, 6. How many ATPs does that represent? 3. Remember I gave you that equivalency earlier on. I said that one reduced NADH has the potential energy to make 3 ATP. Now you see where that comes. Why does reduced FAD produce less? Okay, Caleb here reduced FAD, right? Okay, you want to get rid of yours. It skips these two steps. FAD reduced has lower free energy than reduced NAD. So it donates downhill to Ben. And then the transfer takes place, and now we only get 4 protons pumped out. We missed this first pumping step, if you will. When those 4 protons flow through the ATPase channel, that gives you 2 ATP, right? Eventually, what happens to those electrons that are passing down the chain? I guess I kind of missed the electrons. They're going to go from here to here to here, and eventually they're going to be picked up by oxygen. But to reduce oxygen to water, you need not only electrons, you need protons. Where do they come from? More dissociation of water. So Cody would donate some protons to these electrons. They'd attach to oxygen, then we make water. So you see where the electrons have gone and the path the electrons have taken. They started out in glucose. And then they moved through the citric acid cycle. And then through the electron transport chain. and ultimately ended up in water molecules. And along the way, energy has been trapped by both substrate level phosphorylation and oxidative phosphorylation that makes the necessary ATP that we need. If we run out of oxygen, if all the oxygen becomes reduced into water, then the electron transport chain can't donate any further. So this becomes reduced, this becomes reduced, this becomes. So now your reduced NADs and FADs can't be recycled. So then the citric acid cycle stops. You have to ferment. At least make some ATP. And now you're not pumping hydrogen ions any longer. So you're not making ATP by oxidative phosphorylation. Okay? That make sense? Does that help to kind of put it in context a little bit? Okay. Well, I showed you last time an illustration which is hand-drawn. And if you haven't guessed by now, art is not one of my strengths. But this particular illustration is a good summary of what we just talked about. So let's run through it real quickly here. You can just, you can throw those up in front when you're on your way out. Okay, so this is a crudely drawn mitochondria. So here is our outer membrane. Here is our inner membrane space right here. Here is our inner membrane. And then here is the matrix. This represents the electron transport chain. Compounds that are hydrogen donors and acceptors, and compounds that are electron donors and acceptors. Within the matrix, we're producing all of this reduced NAD and FAD by the citric acid cycle. And the electron transport chain is going to function to recycle those coenzymes so the citric acid cycle can keep going. So again, when reduced NAD donates its hydrogens to the first carrier, it gets recycled. But when that tries to give those hydrogens to this carrier, it'll happily take the electrons, but not the protons. And so protons are being pushed out into that inner membrane space. Those electrons then will have to move to the next carrier so they can accept more from this carrier, so it can accept more from reduced NAD. So the electrons go over here, but it needs some protons. They come from the dissociation of water. And so electrons are flowing through the electron transport chain on their way to oxygen to make water. For every reduced NAD that moves out here, or goes through the system, 6 protons are pumped out here. For every reduced NAD, 4 protons are pumped out here. And then when you go through the ATPase channel, for every 2 protons that flow through the channel, you make an ATP. So this kind of shows you how many protons are being pumped and the total number of ATPs that we can get potentially. Now, there's a little wrinkle in this, what makes it a little bit more confusing. I told you that 38 was the maximum amount of ATP that could be produced per glucose by aerobic cellular respiration. In reality, it is somewhere between 36 and 38. What's the discrepancy there? I'll come back to this slide in a second because this is just a nice cleaner version. Well, let me show you this one right now. This is a cleaner version of what I just showed you. The only thing I don't like about this particular slide, and if you have your PowerPoints here, you might want to make this notation, is they're showing a single proton being pumped out at each of these steps. It's actually 2, so there's 2 pumped out here, 2 here, 2 here, and then for every 2 that go through the channel, we make ATP. Okay? I don't know why they left that off. So let's go here. This is kind of our accounting that we're going to be doing now, to get to this magical number of somewhere between 36 and 38. Okay? So what are we doing? Well, in glycolysis, we're producing 2 ATP by substrate level phosphorylation. However, we're also producing 2 reduced NADs. Those have got to be recycled. If we recycle them through the electron transport chain, how many ATPs would that represent? It's equivalency. Every reduced NAD makes how many ATP? 3, right? So that would represent, you would think, 6 ATP. But there's a little wrinkle. We'll come back to that in a second. Okay, pyruvate in the presence of oxygen will be transported into the mitochondria. The acetyl-CoA will enter the citric acid cycle. And remember, we're accounting on per glucose basis. So 2 acetyl-CoA are produced for every glucose molecule. 1 enters the cycle at each time. So the net total of ATP produced by the citric acid cycle per glucose is 2 additional ones. And that's still substrate level phosphorylation. Okay? But we're also generating 2 reduced NAD in that transition reaction, 6 of them in the citric acid cycle. And I usually lump these together when I do the accounting. I consider the transition reaction to be part of the citric acid cycle. We make 2 reduced FAD. Okay? So right now, what have we got? We've got 8 reduced NAD. Each one of those can produce 3 ATP by oxidative phosphorylation. So that's 24 ATP. We've got 2 reduced FAD. They can only produce 2. That's 4 ATP. But we've got these 2 reduced NADs from glycolysis. So that's 34 ATP. And if you add that to the 4 you get from substrate level phosphorylation, that's where you get your 38 total. But here's the rub. The mitochondrial outer membrane is not freely permeable to this reduced NAD. So what has to happen is the reduced NAD in the cytoplasm has to donate the electrons to a shuttle, a molecule in the membrane, which then passes them off to oxidized coenzymes in the matrix of the mitochondria. And there's 2 kinds of oxidized enzymes. There's oxidized NAD and oxidized FAD. And these can be transferred to either one of those. It just kind of happenstance which one it happens to do. So if it transfers all of its electrons to NAD, then you get that 6 ATP. But if it transfers them to reduced FAD, you only get 4. You get 2 less. That's why there's a discrepancy, and we say somewhere between 36 and 38. Is the fact that these are being made in the cytoplasm. The electron transport chain is in the inner membrane of the mitochondria, and the membrane of the mitochondria isn't freely permeable to those reduced coenzymes. So it shuttles them off and transfers them to coenzymes in the matrix. Okay? That might seem like kind of an esoteric little deal, but I think it's important to explain that because otherwise you see this and you go, why 38 sometimes, why 36 other times? This slide just gives you a representation of how we believe that the ATPase enzyme works, that catalyzes the reaction of ATP formation. And don't get hung up on all of this detail right here about stocks and catalytic sites and knobs and stationary knobs and stators. Basically, this is kind of a little motor-like apparatus, and as the hydrogen ions flow through this ATPase channel protein, energy is harvested to use to make ATP. That's really about all you have to know at that particular point in time. This is just another representation of what I just told you. Okay? This is just the same accounting procedure as was shown in the previous slide. Some people from past experience like this one better than the other one, just visually. It's easier for them to understand. But it contains the same information. This is just a little summary of the energy production from cellular respiration. And this is something that you should be able to work your way through and understand. So that's a good steady aid for the exam. Okay, one final concept that we need to talk about regarding cellular respiration. Cellular respiration has a very important catabolic role. And the catabolic role of cellular respiration is to completely oxidize the glucose. So we form carbon dioxide and water. And to produce as much energy as we possibly can. That's what we've been concentrating on for the last couple of days, is the catabolic role of cellular respiration. But as it turns out, cellular respiration has an important anabolic role as well. Cellular respiration can provide what we call carbon skeletons, partially broken down glucose molecules, which can be used for the biosynthesis of other molecules. And that's important too, because cells need to grow, divide, repair themselves, and to do that they need proteins, carbohydrates, lipids, nucleic acids, because that's what they're made up of. Where do they get those things? They can synthesize many of these molecules out of glucose as a starting material. Glucose is kind of like the plastic of cellular metabolism. Plastic can be manufactured into all kinds of different things. Well, glucose can be arranged into all kinds of different molecules too. Before we talk about the biosynthesis aspect of it, let's talk about another aspect of catabolism. And that is glucose the only molecule that can produce ATP by cellular respiration? Well, no. We already talked about the fact that carbohydrates can be converted into glucose, which can then go through cellular respiration. But what about molecules like proteins and fats? Can they provide energy that can be tapped by this process of cellular respiration? Yeah, they can. How? Well, what's fat? What are fats made up of? They're made up of glycerol and fatty acids. And if you digest fats and break the bonds and release the glycerol and fatty acids, what happens to them? There are metabolic sequences that can occur that can convert glycerol into this compound, glyceraldehyde 3-phosphate. And this is part of glycolysis. So fats can basically feed into glycolysis and then be converted into carbon dioxide and water and produce ATP. Fatty acids can be converted into acetyl-CoA. And acetyl-CoA feeds into the citric acid cycle. Okay? What about proteins? Well, proteins can be hydrolyzed into their amino acids. Amino acids can be deaminated. They can have their amino group removed. And when that happens, some amino acids, when they're deaminated, make pyruvate. Oh, okay, now we're in this glycolysis and citric acid cycle. Some amino acids, when they're deaminated, can be converted into compounds that are part of the citric acid cycle. Alpha-ketoglutarate, fumarate, succinate. Now, notice that the ammonia that's produced by this deamination process could be a problem. Ammonia is toxic to cells. And so cellular metabolism usually has some mechanism to get rid of this ammonia that's produced as we catabolize proteins. We make things like urea, and we secrete that out of the body in our urinary system. So there's a lot of ways for various complex organic food molecules to basically feed into this process of cellular respiration. So this is kind of a central processing pathway, and a lot of different molecules can feed into this generalized pathway. Well, okay, now let's take a look at anabolic roles. Let's say, you eat a whole bunch of sugar and then you plop down on the couch and watch a lot of NCAA games. Okay? No, that's never going to happen. So what's going to happen? Sugar's going to be burned up. You're going to start producing a lot of ATP. And eventually, you're going to have all this ATP. And you're not active, you're not using it for a lot of cellular work like movement. So should you keep producing more ATP? Probably not. Okay? Why burn up your food, your fuel, and make ATP you don't need? So as it turns out, there are some key enzymes in glycolysis that are sensitive to levels of ATP and ADP or AMP. These are two kind of equivalent things. And ATP, excessive ATP, tends to inhibit some of these enzymes of glycolysis. It's called feedback inhibition. Okay? So the product of the pathway, ATP, inhibits more formation. ADP or AMP levels are high when ATP levels are low. So if there's a lot of this, that actually stimulates glycolysis. So if you've been eating a lot and haven't been active, glycolysis is making a lot of ATP. And that can inhibit these enzymes of glycolysis. And instead, what happens to some of these compounds? Instead of glyceraldehyde 3-phosphate being further broken down to make more ATP, what happens? Cell metabolism adjusts. And this reaction now becomes stimulated. And glyceraldehyde 3-phosphate is converted into glycerol. And acetyl-CoA is converted into fatty acids. And these two combine to make fats. Okay? Well, fats are important. Okay? We need to make new fats occasionally. As cells divide, we repair membranes and things like that. So instead of breaking all of our food reserves down to make ATP when we don't need it, we can use those as building blocks or carbon skeletons to make new materials for new cells. That's the anabolic role of cellular respiration. And the balance between catabolism or breakdown and anabolism or synthesis of new molecules can be regulated by things like ATP levels or ADP levels in the cell. Okay? So it gets pretty complicated. And this is just scratching the surface of things. But this gives you some kind of an idea of the importance that enzymes play in regulating something as complex as metabolism. A lot of the stuff that we've talked about, particularly this process of chemiosmosis, is going to carry over into the next material that we cover after this exam, which is photosynthesis. Before we wrap up, there's one thing that I want to make sure that you understand the difference in. I thought it was on this slide. Maybe it's not. Okay, here. Sometimes it's difficult for students to understand the difference between the concept of oxidative phosphorylation and chemiosmosis. And this illustrates it pretty well. Oxidative phosphorylation refers to the whole ball of wax, basically, of how ATP is made, and includes the activity of the electron transport chain, which generates the hydrogen ion gradient, and the process of tapping the hydrogen ion gradient to make ATP. Using the hydrogen ion gradient to make ATP is chemiosmosis. Chemiosmosis and the electron transport chain working together make up this process of oxidative phosphorylation. So see that distinction. Oxidative phosphorylation refers to the whole thing. Chemiosmosis just refers to this part of making ATP using the hydrogen ion gradient. The electron transport chain is what generates or forms the hydrogen ion gradient. So I would encourage you prior to Thursday, if you want to get something out of Thursday, to try to do some review on this stuff. And again, remember, a lot of the important information about this can be obtained by going all the way back to that early slide that asks these kind of fundamental questions about those three pathways. Okay? That's a good thing for you to study. I know Austin said when he did mentoring last night that this is one of the things he spent some time going over. So you should be very comfortable answering those particular kind of questions. So, if you don't have, anybody have any final questions right now? This is unusual. I'll give you a little break today. Anything for lab? Like me on time? Want to come early? Okay. I will send you a study guide for the lab quiz that you'll have next week. I will send that to you this afternoon. So that'll come by at noon. Okay?
Is Austin doing like another review on Thursday?
Yes.
Review night?
Yep.
Is that at 9 or 8?
8. Thursday's 8. Okay? We'll see you Thursday.
Answers to the practice.
One of them has answers on it. One of them doesn't.
Is Austin gonna have it?
Pardon me?
The mentor, Austin, is he gonna have the answers to the ones that don't?
He can help you with the answers, yes, to the one that gets them. Yes,
Can
well,
you print it off real fast? Because I forgot.
yes.
All right. That's only about 45 minutes.
How does ATP get from the matrix and mitochondria into the cytoplasm? There must be transporters
Yeah.
because it's such a big and charged molecule.
Yeah, yeah.
The other thing that I think would be interesting, I may try to figure this out. Yeah.